Thank you to you, and thank you for the invitation, and thank you, everybody for being here today. I'm very excited to present this ongoing effort, my postdoc work, trying to understand how neuron diversity emerges. It's a question that drives me. What are the cellular and molecular mechanisms that happen during development that generate this humongous neuron diversity that animals show in nature. We're all familiar with these drawings by Cajal, in which you look at different neurons, and neurons have different shapes. Some of them have bigger, some of them are smaller, some of them have more dendrites, some of them have fewer dendrites. And then, of course, now we know that morphology is one of their aspects, but also they have many other properties that are generated during development, right? So not only the morphology, but also their physiology, and then things that they express. We also do know that two neurons that look very similar in morphology can have a different physiological properties, or express a completely different set of genes, or express very similar set of genes and have a completely different morphology, right? So it's not clear what the relationship between all of these characteristics are, and then how they are generated during development. To make things even more interesting, different animals have found different solutions to this problem of generation of neuron diversity. Some animals do it in what is known as a deterministic way, meaning that you can sort of, with confidence, you can predict when and where a specific kind of neuron is going to be generated, and which transcription factors are deployed to do so. But then once you get to things like vertebrates, it's not necessarily true that things are done deterministically, and then things become very big. So studying this process in totality and it's a system-wide level, it becomes very burdensome. So then answering these three questions that are in blue, that are my main interest, in vertebrates, becomes very complicated, right? How is this created at the molecular level? What are the logic that generate this or that molecular subtypes? What is the relationship between molecular and cellular behaviors and the generation of diversity? How is it? Because it's intrinsically very dynamic, so it's not, it's really not understood how this happens. And then how does this happen with robustness? How could it be that everybody has different amount of neurons in their brains, but on average we're all the same, right? So how does this happen in with robustness between animals, but also within the same animal, that each layer and each compartment has a specific kind of neuron generated. So in order to understand this question and to tackle this question, I'm using a population of neurons that zebrafish have in the spinal cord called Rohan-beard neurons, which are somatosensory neurons, which means that they detect peripheral stimuli. And to do so, they have these beautiful projections that they send to the periphery. They're born really early, and they're also active really early. There's only a handful of them per animal, so around like 150, 100, and 70 per animal, which is very amenable for things like counting, or manipulations and so on and so forth. And the reason why I study them is because fishes and frogs have these very interesting property that, unlike us, we have only one somatosensory system, which is the DRGs in the right next to the spinal cord. But fishes have two, classically, it's described that they have two somatosensory systems during development. One that is the Rohan-beard neurons, and then people thought that they disappear for by parent cell death, but then I sort of by accident found that this is not true. So the Rohan-beard neurons remain during life, and then coexist with the DRG. But it also means that I can study the Rohan-beard neurons to understand the development of the somatosensory system, because in the end they serve a similar purpose, which is proprioception, mechanoreception, nociception. They do this because they express specific molecules, dedicated molecules to do so. But until now, people don't know in totality what these are. So what I did is to do single cell during development of the rhomboid neurons. And then I was very happy because I found a lot of known transcription factors that people have published for the, like to be important for the development. But I also found a lot of new transcription factors and a new, for instance, combination of signaling molecules, right? So the more heterogeneous of the transcriptome than what people previously thought. They're not only heterogeneous in the transcriptome, but also once I started looking at when and where things are expressed, I saw that while there are some functional channels that seem to be expressed in every single rhomboid neuron, some of them are in a few of them along the AP axis, and then some of them seem to be restricted to either the anterior part or the middle part or the posterior part of the trunk. So, I don't know whether this is physiologically relevant or not, but this is another layer of complexity. And the interesting thing is that this is also heterogeneous even between individuals, right? That, so they're not necessarily exactly the same. So then how is this built? So in order to understand how this is all built, I teamed up with a postdoc in the laboratory, Yinan, who developed this whole mount spatial transcriptomics technique. And then it means that instead of sectioning, you can take just a whole larva, clear it, and then hybridize it for as many genes as you want. It conserves the shape and the position, which means that you can see where the cells are, but you can also quantify individual mRNAs. So you can do this for, like I said, for as many genes as you want, and then for as many rounds as you want. And then in the end, you can have there should be there should be a summary here. But anyway, the summary is that if you take everything all the rounds and then you summarize it, it will look like something like this, which I call the Russian doll model of diversity, which means that there are some channels or some molecules that are in all rhomboid neurons. There's a major split between two mutually exclusive categories, NTRK2 and NTRK3, and then within those you find specific subtypes. But then how do you generate this diversity? And in order to understand this, I looked at the development, and interestingly, this diversity doesn't explode all at the same time, so it happens in a staggered model, which means that at the beginning you don't have functional channels. Then a little bit later, you have some, but not all functional channels. And then in the end, you have most of the functional channels. So to understand how this is generated, I look at the development of the rhomboid neurons, because these neurons are born during gastrulation, but we don't know their lineage relationship, right? Well, we don't know whether these two that are adjacent are siblings or cousins, so they have nothing to do with each other. So in order to understand this, I did light sheet imaging of the process of rhomboid neuron development, and then I saw that they have extremely simple lineages, right? So either they post-mitotic already during gastrulation, or they divide, once or twice. They arise as independent units, which means that the progenitor doesn't divide and form two rhomboid neurons. They move coherently, and they don't switch positions. So then this made me wonder what the molecular subtype of each one of these were. So what I did to tackle this was I took this animal, I imaged it, I fixed it, and then I did a spatial transcriptomics on top of it. And then what I saw was that, for instance, cell division is not a predictor of this major split between 2A and 3A, for instance. And in fact, you can block proliferation and you still have this split between the two. So then what determines this split? So I went and looked earlier, because this split of mutually exclusive categories is not always like this. So at the beginning, all of them are 2A, and then some of them flip to 3A, and then this gets resolved. But then the interesting thing is that the number of the rhomboid neurons that flip to 3A is almost 50-50. It's not 50-50, but it's almost 50-50, which means, or may mean, that this is a stochastic process. So to understand how this process happens, I underwent a CRISPR screen for many, many transcription factors, and today you're going to see just two that affect this split between the 2A and the 3A. So if you mutate ILED1, very important transcription factor in sensory neuron development, they do develop and they do mature, but up to a point. But then this is split, so you get fewer of the 3A. And another set of transcription factors co-expressed with ILED1 is PO4 factors, which are in all rhomboid neurons, also not in motor neurons. And the interesting thing is, if you make mutants for PO4F1 and F4, In a wild type, which will have 4 copies of these PO4s, they all split, 55-45. If you remove 3 copies, they still flip, but in fewer cases. But if you get rid of all 4 copies, they don't flip, which means that probably means that there's a dosage sensitivity for the process. So the model now is that you induce the Rohambi neurons, and then they start the development program. And then these 3 transcription factors, we still don't know how, are sufficient to make some of them express RUNX1. And this kicks in a program of the 3A, NTRK3A, and then RUNX1 goes away, and then RUNX3 takes its place, and then it sort of locks in the fate of this 3A. And then within that one, you have the patterning, which I call the spinal cord program, which is basically this TRIP and ADCF1A that you saw. So everything that I told you today is summarized here, but basically Rohambi neurons use transcription factors in space and in time to generate this diversity of subtypes in space and in time. It doesn't seem like in other places this generation of diversity is linked to cell lineage. It occurs in the postmitotic neurons. And this robustness is achieved by a product of having the right proportion of transcription factors in space and in time to, basically take a population of neurons and segregate them into a mix of subtypes. So with that, I would like to, thank you all for being here today, and thank my laboratory, especially Alex, for being a fantastic mentor, and then everybody in the Shear laboratory and everybody who's participating and helping me with the tracking, and then our collaborators. And with that, I will be very, very happy to take any questions that you have. Thank you.
All right. Thank you, Joaquin, for exciting results, exciting observations. This is really extremely cool that you can do spatial transcriptomics in the whole mount. And yeah, that looked really sharp, the images, and yeah, really great. We are still waiting for the questions in the Q&A box. So please don't hesitate to, participants, please don't hesitate to type your question. We have some time for that. And maybe while we are waiting, I will ask also a question that interests me. So before you described that maybe this is some celadon. So at first I thought maybe some of the progenitors of the Rohamberg neurons already get primed, because you say they
you're asking whether there's priming.
They develop independently and then just have 1 or 2 divisions, and some of them will decide to go to one class of neurons or the other class of neurons. And then I thought maybe like it is already primed before even the Rohamberg neurons emerge.
It is possible, but like so I started imaging relatively early, which means that I'm basically at the beginning of gastrulation, and then at the beginning of gastrulation there are not that many signals that you could say are relatively basically the embryo is still trying to decide what the head is, what the tail is, what the spinal cord is. There is still no neural plate, so that happens a little bit later. So it is possible that there is a signal, like non-cell autonomous signal, but I didn't show it today, but I do have some data showing that if you do disrupt the neural plate and have fewer Rohamberg neurons, they still segregate into these 2 major subclasses. So it is possible that they talk to each other, but I don't think that it's because there are cells in between. So it could be that there is a signal from the cells surrounding them, but I have, basically, like I said, like I didn't show today, but like I think it's probably cell autonomous, which is very, very interesting.
Okay. Yes. Thank you. Yes, it is really quite interesting. So there are a few questions, but I think for the first question, it is sort of similar to the question that I just asked. The next one is, Hi, Joaquin, beautiful work. The images are amazing. Have you been able to look at the binding sites for the transcriptional factors? I'm wondering if the dosage is coming from more transcriptional fighting binding on one side, or increased amount of transcriptional factor means they bind to more than one place.
That is an excellent question. I have actually, and so the RunX1 promoter does have binding sites for islet and PO. They are very close to each other. The interesting thing about RunX1 is bizarre because RunX1 is very close to the telomeres. Until recently, we didn't know exactly how long the gene was, but now we know that it is. So it has two isoforms. So like what will be the short isoform, and then 100 kilobases away there is a second promoter. So it could be that it's related to this other node. Like that's basically what I'm trying to find out. And then now that I'm in the job market, like it's basically what I'm trying to propose that I do, right? How do all these transcription factors do it?
Exactly, like regulatory logic.
Sorry.
Like a regulatory logic.
Yeah, or that, for instance, islet 1 can also open chromatin. So, maybe islet 1, what it does is just to facilitate, the chromatin open, and then the dosage of the PO factors is just basically the old, bind into the promoter and then give PO2 robustness to go and activate it, right?
Yes. So maybe a very short answer to the last question. Do you have any thoughts on environmental inputs like flow or something like mechanical cues that dictate the fate and molecular heterogeneity in these neurons?
It could be, yeah. I mean, I've looked at hippo mutants, which is like one of many, but like hippo mutants have this segregation fine. They develop all the cell types, etc. Raw homogeneous form, etc. I do not know. So I haven't looked in many other things. But for instance, piezo, like if you make piezo mutants, they still segregate. So it's not piezo that because it's a mechanosensory channel that they express like really early. But yeah, I haven't looked at like how viscous, for instance, the neuroplate is that may explain this one becomes this subtype, and the other one becomes the other subtype. But it's super interesting. I mean, I agree that it could be something like that.
Yeah. All right. Many thanks.
Thank you.
I hope you continue your work and really can dissect this logic. But super exciting and definitely exciting methods. Many thanks.